So we will now discuss about the motifs in protein with respect to gene expression. Now motifs are actually patterns, the patterns which are found in the supersecondary structure of proteins, which perform certain special function. The main basis of the function of that protein may be. Now, most of the motifs are actually DNA binding motifs. So, what DNA binding motif is? They are the certain patterns found in the proteins, mainly transcription factors, as written by TF here, which helps to interact with the DNA and carry out certain processes. So, motifs are actually structural patterns found in the proteins which perform some specific function of that protein. For example, the DNA binding motif, they are found in certain, most of the transcription factor they contain this type of motif, DNA binding motif, so that they can able to interact with the DNA and carry out certain processes. Okay, so certain motifs in protein mediate the binding of regulatory protein, that is transcription factors, to the DNA. The specific control of transcription occurs by binding of regulatory proteins with high affinity to the correct region of DNA. So there are certain motifs in proteins that mediate in the binding of regulatory proteins, activators, repressors, right? Transcription factors are generally activators. Okay, they are recruited by activators, most of them, to the DNA. And the specific control of transcription occurs by binding of regulatory proteins with high affinity to the correct regions of the DNA. These structural motifs are found on many different proteins, and most of the structural motifs are found on transcription factors in eukaryotes. So, most of the structural motifs are found in different proteins, and transcription factors have a DNA binding motif that you know just now. We know that in order for transcription initiation, we need the minimum requirement of general transcription factor and RNA polymerase, right? For transcription process to initiate, we need RNA polymerase, but RNA polymerase itself cannot able to bind. For this we require some transcription factors, and these transcription factors have this type of motifs so that they can interact with the DNA at specific site. You know where is that specific site in the promoter, and carry out the initiation process. Apart from general transcription factors, there are some other transcription factors known as specialized transcription factors, which binds to the specific regions of the DNA and recruits your activators or repressors. In the previous video we have studied that what are activators or repressors. Activators which induce or which stimulate the transcription factor to go for transcription process, whereas repressor will prevent the transcription of that specific gene. So apart from general transcription factors, there are some other transcription factors known as specialized transcription factors, which binds to the specific regions of the DNA and recruits your activators or repressors, and thereby regulating the transcription of your DNA. Okay, so these are actually the motifs, the general definition of motifs. Motifs are actually patterns found in the supersecondary structure of protein, which appears in a variety of other molecules. Most of the motifs may be a DNA binding motif, and what a DNA binding motif is, DNA binding motif are certain patterns found in proteins. Mainly it is found in transcription factors, which helps to interact with the DNA and carry out the certain processes like like you're stimulating the transcription process, inducing or allowing the transcription process to begin, maybe like that. uh, Now, the structure of motif are generally found on different proteins. These structural motifs are found generally on different proteins, and most of the structural motifs are found in your transcription factors in eukaryotes. We know that in order for transcription to begin, we need minimum requirement of your transcription factors and RNA polymerase, generalized or general transcription factors that are normally required for transcription process to begin. Apart from general transcription factors, there are some other transcription factors called specialized transcription factors, which binds to specific regions of the DNA and recruits your activators or repressors for regulating the transcription. Generalized, there are some specialized transcription factors which binds to certain regions of the DNA with the help of their DNA binding motif, and then they recruit the activators or repressors and thereby regulating the transcription. Now a question arises in our mind, maybe arise in your mind, that how transcription factor binds to the DNA is a very important, it is a question of interest. It's due to the help of their motif. Okay, there are some patterns in the transcription factors that helps to bind to that particular site in the DNA, so that we will discuss on the types of motif that are found normally. So in transcription factors, there are specific DNA binding motif. In transcription factors, there are specific DNA binding motifs which ensures the specificity of that specific transcription factor to bind in a sequence specific manner and thereby activating or repressing transcription. Let us zoom into it, if you can able to see now. So in transcription factors, there are specific DNA binding motifs, right? There are specific DNA binding motifs which ensures the specificity of that specified transcription factor written by TF to bind in a sequence specific manner and thereby activating or repressing your transcription. So normally the transcription factor have a specialized DNA binding motif which able to interact at a specific region in the DNA, in the major group or in the minor group, and then it will allow recruitment of the activators or repressors. Okay, it will allow recruitment of the transcription factors, other transcription factors, to bind and carry out the transcription of the gene. Now the main important thing, okay, one more thing is your this one. It is saying that the tertiary structure of the protein, the tertiary structure of the protein belonging to the different kinds of structural domains and structural motifs. Again I repeat, the tertiary structure of the proteins belonging to the different kinds of structural domains and structural motifs. If you look at the tertiary structure that you have learned in the first semester in biochemistry classes, it shows that when protein fold into tertiary structure, the secondary structure, you know, the alpha helix, the beta pleated sheet, the beta turn, parallel and anti-parallel beta sheet. They also again folded to form the tertiary structure, to form domains and motifs. So the tertiary structure of protein belonging to different kinds of structural domains and structural motifs. They are composed of structural domains and structural motifs. Who are composed of structural domains and structural motifs? When the proteins fold into tertiary structure, right? Structural motifs means interaction between all their secondary structure. If you say simplicity, that what are motifs here, I have said motifs are the patterns found in the protein which are able to do certain function. So the structural motifs means your interaction between all their secondary structure, like the alpha helix interacting with beta sheet, the alpha helix interacting with another alpha helix, or a beta sheet with another beta sheet, and loop and turn. So the interaction between them creates a specialized structure called motifs which are able to do a certain function. And the best example is your DNA binding motif, which that motif will helps to interact with the DNA at specific sites and recruit the activators or recruit the transcription factors to follow the transcription, or it may regulate the transcription. Now comes the main types of DNA binding motifs, because these are the main things that you will normally study. There are many other types of motifs are also found, but these are the structural DNA binding motifs. These motifs are only meant for interaction with the DNA actually. Okay, the first motif is your helix turn helix motif. In short, it is called HTH. Then comes your zinc finger motif, because there is a presence of zinc in that motif. Leucine zipper motif, as the name suggests, there is a leucine amino acid maybe, and zipper, its action is look like a zipper, that's why the name come. And helix loop helix motif. Remember, these most of the structural motifs are found on the transcription factors, so you need not to worry, most of the transcription factors have this type of motifs. Okay, so first I will begin with helix turn helix motif, or HTH. Let us see what it has been saying. Helix turn helix motif, or HTH motif, has two alpha helix followed by a beta turn, and it binds to the major groove of DNA. Very important. The first point is very important. Helix turn helix motif is made up of two alpha helix. It is made up of two alpha helix followed by a beta turn, and binds to the, this motif can able to bind to the major groove of DNA. Okay, come to the next point. Helix turn helix motif is about 20 to 25 amino acid long. Okay, helix turn helix motif is about 20 to 25 amino acid long. Okay, this you will easily understand this point. Helix turn helix motif bind to the major groove of DNA as given in the early point. Helix turn helix motif binds to the major groove of DNA through various interactions like your hydrogen bond, van der Waals interaction, right? So it is saying that this helix turn helix motif is about 20 to 25 amino acid long. It binds to the major groove of DNA. It binds to the major groove of DNA by various type of interaction. What are the types of interaction? Like your hydrogen bond, like your van der Waals interaction. Okay, now it has been said in a very small point. Let us see if you can able to see this. The point where this helix turn helix motif, the point where helix turn helix motif interact with the DNA at the major group, at this point the amino acid residues of the helix turn helix motif bind to the specific bases in the DNA. Again I repeat, the point where your helix turn helix motif interact at the major groove of DNA. At this point, there are the specific amino acid residues in the helix turn helix motif able to bind to the specific bases in the DNA to carry out that particular function, maybe to bind, right? So these helix turn helix motifs that are found in most of the transcription factors, they can interact with the major groove of DNA at the point, and on that point where it interacts, the amino acid which are found on helix turn helix motif, they bind to the specific bases in the DNA, right? Now comes the next point. Let us see the residues, the amino acid residues short form I have written a bar symbol. So these amino acid residues that are exposed in the major groove from the helix turn helix protein motif, they actually bind to specific bases in the DNA. As stated earlier, just I have said, the amino acid residues that are exposed in the major groove from the helix turn helix protein motif, they actually bind to specific bases in the DNA. Lambda repressor, which is an important protein, used for lytic and lysogenic switch in case of bacteriophage, is also having the same helix turn helix motif. Remember, bacteriophage are those phage viruses that can kill the bacteria, and it can switch between lytic cycle and lysogenic cycle. Okay, so how do bacteriophages switch from lytic cycle to lysogenic, again lysogenic to lytic cycle? This switching is controlled by lambda repressor, which is a very important protein used for switching from lytic to lysogenic and vice versa. This lambda repressor also having this type of helix turn helix motif. So helix turn helix motif function is pretty much conserved from bacteria to human. These motifs are found in bacteria, in virus, in human also, right? So this helix turn helix motif is pretty much conserved from bacteria to human. So by observing the sequence and comparing the sequence, we can find that they are most of them conserved. Okay, more to go about the helix turn helix motif. Okay, another important class of transcription factor known as homeodomain containing transcription factor are highly conserved and encoded by 180 base pair of DNA. That is, if you divide it by 3, it will be 60, because 3 nucleotides make a codon, and so it will be 60 amino acids. Okay, so another important class of transcription factors known as homeodomain containing transcription factors, they are highly conserved and are encoded by 180 base pair of DNA. That is, it is made up of 60 amino acid termed as homeobox, have also that same type of helix turn helix motif. Again I repeat, another important classes of transcription factors known as homeodomain containing transcription factors. These homeodomain containing transcription factors have a homeobox. They are highly conserved. They are encoded by 180 base pair of DNA. That is, it is made up of 60 amino acid segments termed as homeobox. These homeobox also having that same type of helix turn helix motif, but instead of 2 helix, it has 3 helix. Remember, I have said just now, normally helix turn helix motif have two alpha helix followed by a beta turn and binds to the major groove of DNA. But in case of this example, that is your homeodomain containing transcription factor, they are highly conserved and encoded by 180 base pair of DNA, that is, it is made up of 160 amino acids known as homeobox, and this homeobox have also similar structure of helix turn helix motif, but instead of 2 helix it has 3 helix. The homeodomain containing transcription factor have 3 helix instead of 2 helix, right? This helix turn helix containing homeodomain containing transcription factor are important for segment polarity patterning patterning of both from the fly to a mammalian embryo. So this homeodomain this helix turn helix containing homeodomain containing transcription factor, they are important for segment polarity. Okay, they are very important for segment polarity and patterning of both from fly to a mammalian embryo. So it is very conserved actually. It's homeodomain containing transcription factor. It is important for segment polarity. Okay, it is important for segment polarity. Now if you want to know about what is segment polarity, I will discuss to you later on in this video. In the description section you can see, okay, what is segment polarity and the patterning of both from fly to a mammalian embryo. How the pattern of the human embryo will develop, the mammalian embryo will develop, the fly embryo will develop. Okay, that will be somehow regulated by these homeodomain containing transcription factors. So this helix turn helix is the domain part, a segment part of the protein which specifically interacts with the DNA. Example of these helix turn helix motif are your lactose repressor, your cyclic AMP is also an example having that not cyclic AMP, again I repeat. So this example of helix turn helix motif is your lactose repressor and your catabolite activator protein that I have just we have just discussed during the prokaryotic gene regulation, that is your lactose operon. There I have said discuss about the catabolite activator protein, cap protein, which normally bind with cyclic AMP to form that complex to allow the RNA polymers to bind to the promoter and carries out the transcription of the lac operon genes. So this cyclic AMP catabolite activator protein also having this type of motifs which can interact with the DNA. Then lactose repressor also having this type of motif. Okay, and several developmentally important transcription factors in animals. Developmentally means related to development, just like I have said here, the helix turn helix containing homeodomain containing transcription factors which are very important for your segment polarity as occurs during the embryo development, right? And patterning from both from a fly to an mammalian embryo. Okay, coming to the next part. So first of all I want to show you about this one, the picture of this helix turn helix motif. The left figure that you can see, it shows a ideal structure of your helix turn helix motif. Okay, two alpha helix with your turn, right? Two alpha helix with your turn. So you can see two alpha helix has been shown in barrel cylinder shape. Okay, and we also see a turn that has not been shown too clearly, but I have marked it, amino acid sequence from a turn. Out of these two alpha helix, you can also able to see in the right also. I have to forgot to mention you have to also see the right figure also, that this helix turn helix motif can also be made up of many alpha helix, not only two but also much more than two, as you can see in the lambda repressor protein. The lambda repressor protein also contains helix turn helix motif, but it contains almostly 5 alpha helix connected by a beta turn. I have named them from 1 to 5 depending on where the amino terminal end starts and where the carboxy terminal end stops. Okay, so this is the general structure of your helix turn helix motif. Now the what I want to show you is your recognition helix. So out of these two helix, you can see in the helix turn helix motif, one of the helix is called recognition helix, and recognition helix is actually involved in identification of the target DNA sequence. If you can able to see, I am zooming it more. This recognition helix involved in identification of the target DNA sequence. Okay, so come to the now the point. So you can able to adjust this point with the diagram now. So one of the two alpha helix is known as recognition helix. As I have said, one of the two alpha helix is your recognition helix, which is involved in identification of the target DNA sequence. That means this recognition helix involved in identification of the target DNA sequence. Okay, each of the two helix are usually 7 to 9 amino acids long and are joined by a short stretch of polypeptide up to 20 amino acids long. So each of these two helix is about how much? 7 to 9 amino acid long, and it is joined by a stretch of polypeptide up to 20 amino acid. So this stretch of polypeptide is up to 20 amino acid, which has been shown in short here. The actual figure is look like this. 20 amino acids are involved in the turn. Okay, here. Okay, next, the spacer polypeptide which lies between two alpha helix has a beta sheet type structure. The spacer polypeptide, the space which lies between the two alpha helices, has a beta sheet type of structure. The protein with this type of motif are active in dimer formation. Now this is important, that those protein maybe your transcription factor or some regulatory proteins, those protein which have this type of helix turn helix motif, they are active to form dimer. They will be active to form dimer. Dimer means two types of proteins are required to activate that function. So the proteins with this type of motif, those proteins that are having this helix turn helix motif, mostly these proteins are active in dimer formation and always interacts with the DNA at the major group at two sites. Two sites means because dimer, it forms a dimer, so it will interact with the DNA at two sites. Okay, that is two similar proteins containing two helix turn helix motifs interacts with the DNA at the major groove at two sites. So two similar proteins containing here proteins that containing helix turn helix motif, they will interact with the two sides of the DNA at the major groove, right? So this was about the helix turn helix motif, right? So most of the proteins which are having this type of motif, they are active when it forms a dimer. Another protein comes, similar protein comes to form having that same helix turn helix motif to form dimer and interacts with the DNA. Okay, now the activity of this type of protein themselves usually regulated by other small molecules. Obviously, maybe some repressor comes and interacts and prevents the activity of that proteins. These control molecules can bind to the regulatory proteins in an allosteric manner and changes its conformation. As a result, the distance between two recognition helices can be changed, which modulate their binding profile with the target DNA. So, the activity of this type of protein themselves is usually regulated by other small molecules. The protein which does this activity, it can be regulated by other molecules, because most of the proteins which have this helix turn helix motif, they form a dimer to become active. Now some regulatory proteins come and interfere their function, that function will be stopped, so it can be regulated by other small molecules. It is saying these control molecules can bind to the regulatory proteins in an allosteric manner. So in an allosteric manner, these molecules which will controlling the expression of these proteins having the helix turn helix motif, they will bind with allosteric manner and changes its conformation. As a result, the distance between two recognition helices can be changed, which modulate their binding profile with target DNA. So this was about the helix turn helix motif. See the figure now at last. So you can see a recognition helix which can interact with the DNA sequence. There is also another helix, but sometimes more than one helix can also be observed, as shown in the lambda repressor protein. Okay, now you can see in this lambda repressor protein that the alpha helix 3 is actually here acting as a recognition helix. Okay, so now I will discuss about the next motif, that is your zinc finger motif. Zinc finger motif is not too much large, it is very short. Let us see what it is saying. Zinc finger is a kind of structural motif that is present in many proteins. So this zinc finger is a kind of structural motif that is present in many proteins. Most of the time, these zinc finger motifs are found in those proteins which are interacting with DNA or RNA. Again I repeat, let us zoom it. Zinc finger is a kind of structural motif. Okay, it is a structural motif that is present in many proteins. It is found in many proteins. Most of the time, this zinc finger motif are found in those proteins which interact with DNA and RNA. Okay, so this type of motif are found in those proteins which interacts with DNA or RNA, like your transcription factors that interacts with the promoter region. Zinc finger motif was first observed in transcription factor 3. Zinc finger motif was first observed in transcription factor 3, that is your transcription factor 3, in short we say TF3, in Xenopas laevis, which is a type of frog. So it was first discovered in transcription factor 3 of Xenopas laevis, which is a type of frog. Okay, I am showing you the structure of zinc finger motif. Okay, so the arrow mark represents the beta sheet. Okay, arrow mark represents the beta sheet. You have already studied the patterns. Okay, these are actually the ribbon shape model. Okay, that you can see. There is also some ball and stick model that you can able to see. Okay, so this is the beta sheet that you can see. From the amino terminal you can see the one beta sheet. Okay, thus there is some cysteine molecules are also, cysteine amino acids are also present here also. Then comes, and at the end, carboxy terminal end, there ends with the alpha helix. Okay, now you can able to see here that within this alpha helix and beta sheet they are interacting with the zinc ion, interacting with the zinc with the help of their amino acids. See the beta sheet having cysteine residues, amino acid residues at certain site, which is interacting with the histidine residues also via the zinc. Okay, so what is saying here, so let us see. If you look at the alpha helix is the place where it interacts with the, binds to the major groove of the DNA, and with the help of zinc, with the help of Zn plus 2 ion, this structural motif is stabilized by interaction of zinc with specific amino acids present in the motif. Again I repeat, with the help of this zinc ion, this structural motif, as you can see, is stabilized by interaction of zinc, interaction of zinc with specific amino acids present in the motif. Okay, so this is the main structural feature of the zinc finger motif. So here zinc Zn plus 2 is stabilizing, is helping to stabilize this motif by interaction of zinc with specific amino acids found in the motif like cysteine and histidine. Zn plus 2 or zinc plays an important role in maintaining the structural fold as it holds all the structures, secondary structures, together by interacting with the amino acids present there, as I have shown to you. So the zinc plays an important role in maintaining the structural fold as it is holding all the structures, the secondary structures, together by interacting with the amino acids present there. So here the zinc is interacting and maintaining the secondary structures by interacting with specific amino acid residues found on this protein. For proper binding with the DNA or RNA, we require a specific type of amino acid only that are present on this motif. For proper binding with DNA or RNA, we require specific type of amino acid only which are present on this motif. Now, depending on the type of amino acid that are present in those structural motifs, it will determine where exactly it is going to bind to the DNA and function. So, for proper binding with DNA or RNA, we require a specific type of amino acid only which are present on this motif. To bind with the DNA or RNA, we need a special type of antibody actually, which are found, which should be present on this motif. Now, depending on the type of amino acid that are present on those structural motif, it will determine where exactly it is going to bind with the DNA and going to function, right? There are 4 amino acid residues which are involved in the zinc binding that you can able to see there. There are 4 amino acid residues involved in the zinc binding. Usually these 4 amino acid residues are 2 cysteine, which are found on the beta sheet and beta turn, and 2 histidine found on the alpha helix. However, sometimes 4 cysteine may also be involved to interact with the zinc to form this type of motif. Based on the amino acid associated with the zinc, the zinc finger is known as cis2 his2 or cis2 cis2 finger. So don't be afraid. Cis2 his2 are actually what? Cis2 his2 are actually those zinc finger motif that actually involve 2 cysteine residues and 2 histine residues interacting with the zinc to stabilize that motif, to stabilize the secondary structure. Okay, whereas cis2 finger are actually 4 cysteine residues which are involving to interact with the zinc. I have not shown the diagram of cis2. I have only shown the cis2 his2. Okay, but there may be cis2 4 cysteine residues involving in some sort of interacting with the interacting with the zinc to form that, to stabilize that structural motif. Okay. Now these amino acid residues are usually present repeatedly at regular interval. These residues that we are talking about, the cysteine, the histidine found on these motifs, found on these proteins, are usually present repeatedly. They are usually present repeatedly at regular interval. The N-terminal half of this zinc finger forms a beta sheet, as I have shown you in the diagram. I will let you to go to the diagram again. The N-terminal half of the zinc finger forms a beta sheet, while the other half forms the alpha helix. See, if you want to see the N-terminal begins from here, and from the beginning of the N terminal your beta sheet is involving here. Okay, N terminal has this beta sheet pattern. Okay, so N terminal half of the zinc finger forms a beta sheet, while the other half forms the alpha helix. The alpha helix can be easily bind to the major groove of the DNA. So these alpha helix, as you can see, these alpha helix can bind to the major groove of the DNA, right? And each zinc finger makes a 2 sequence specific contacts with the DNA. Each zinc finger can make 2 sequence specific contact with the DNA. Within the major groove, there are certain palindromic sequence. Again I repeat, each zinc finger can make a 2 sequence specific contact with the DNA. Now if you want to look at the major groove of DNA, there are certain palindromic sequence. Palindromic means which can be reading same from the 5' to 3' direction as well as from 3' to 5' direction. I want to show you a palindromic sequence here like this. See this palindromic, double A, double T, double A. If you read from 5 prime to 3 prime, okay, as well as if you read from 3 prime to 5 prime, it will be read like same, double A, double T, double A. So this is a palindromic sequence. Okay, there are some names also which are associated with palindromic. For simplicity I want to show you the name, like Nayan. If you write the name words letters from left to right, it will also spell as Nayan. If you spell from right to left, it will also spell as Nayan. So these are palindromic words, or in case of DNA it is palindromic sequence here. Okay, so these palindromic sequence are found in the major group. Certain palindromic sequence are found on the major group of the DNA. So within the major group of DNA, there are certain palindromic sequences within which this type of motifs binds. The cis2 his2 zinc finger are present in a number of transcription factors. The motif, that structure that I have shown to you repeatedly now, this cis2, it is actually cis2 his2 zinc finger. They are present in number of transcription factor, like your glucocorticoid receptor, like your TF3A, like your GAG gene products they contain. So most of the transcription factors have this type of cis2 his2 zinc finger, right? Like your glucocorticoid receptor, like your TF3A, like your GAG gene products. Example of zinc finger motifs containing proteins are your steroid receptors. If you can able to see here, example of zinc finger motifs containing protein are your steroid receptors, your thyroid hormone receptor. Okay, thyroid hormone receptor. Okay, so examples of zinc finger motifs containing proteins are your steroid receptors and thyroid hormone receptors. Now, little bit information has been given about the thyroid hormone receptor. Thyroid hormone receptor having cis2 his2 motif. If you can able to see, I am zooming it. Thyroid hormone receptor is having a cis2 his2 zinc finger motif. So it is a nucleoreceptor, that is this receptor present in nucleus. Thyroid hormone receptor is a nucleoreceptor which is present in the nucleus. When thyroid hormone binds, it activates or represses the transcription by interaction with the co-activators or co-receptors. So repressors. So thyroid hormone receptor is a nucleoreceptor, that is the receptor which is present in the nucleus. When thyroid hormone binds, it activates or represses the transcription. Where the thyroid hormone will bind? It will bind to a receptor, the thyroid hormone receptor, which have this type of motif. So when thyroid hormone binds, it will activate or repress the transcription by interacting with the co-activators or co-repressor. So this was about your zinc finger motif, right? With the help of specific amino acid like cysteine, histidine or cysteine cysteine, they can interact with the zinc and stabilize the secondary structure of the proteins. Okay, and they can interact with the DNA and carries out certain functions, like your most of the transcription factors, like your glucocorticoid receptors, TF3A, GAG gene products having this type of motif. Next comes two more to go, leucine zipper motif. Leucine zipper motif is found in many different proteins like your eukaryotic transcription factor. I have told to you from the beginning that don't worry if you wish to give an example of the structural motifs. Most of the transcription factor have this type of structural motifs. So leucine zipper is found in many different proteins like your eukaryotic transcription factors. Leucine zipper is a dimeric protein. It is a dimeric protein. That means it forms a dimer. It forms a dimer, right? That is two monomer. with two alpha helices that crisscross over each other just like a scissor. See, you can see two alpha helices as shown here that crisscross with each other like a scissor. This part in the middle is your dimerization domain. Okay, here it has also been shown the C terminal end, the amino terminal end. So it is actually two monomers. It is a dimeric protein. One protein, two protein, both protein having N terminal and C terminal as shown here very clearly, and they have crisscrossed with each other to form this type of motif, right? Here it has been shown the different types of amino acids that are found on these two monomeric proteins that are able to interact with each other. Okay, so leucine zipper is a dimeric protein. That means it is made up of two monomers with two alpha helices that crisscross with each other to form a like a scissor, right? And you can see clearly the alpha helices that are found in each of these two monomers, and they crisscross in each other with to form a point. That point is your dimerization domain. Okay, so each of these alpha helix consists of at least 30 amino acids. Each of the alpha helices consists of 30 amino acids. If you look, out of those 30 amino acid, leucine found and repeated at every 7th amino acid residue. This is very important. That's why the name leucine comes here. Each of the alpha helix that we are talking about, it consists of at least 30 amino acid residues, and out of those 30 amino acid residues, leucine is found and repeated at every 7th amino acid residue. This was a longitudinal section. If I show you to a cross section, it will look like this. This first round that you can see, imagine it is a first round of alpha helix. This is the second round of alpha helix. 3.6 I think 3.6 amino acids are involved per turn. I have forgotten. Let me know if you check out this thing that how per turn of alpha helix how many amino acids are involved. I think 3.6 or maybe 4.6. No, 3.6 I think. So let me know. So the two alpha helix has been that the two turns of the alpha helix has been shown. Okay, two turns of the alpha helix has been shown, and you can see here in these two turns the 7th amino acid is your leucine. Okay, and they are interacting with each other. So it is saying in this way that the two alpha helix consist of at least 30 amino acid, and out of this alpha helix that consists of 30 amino acid, leucine is found and repeated at every 7th amino acid residue. It is found and repeated at every 7th amino acid residue. Leucine is actually observed at the dimerization domain. That is the point where the two alpha helix has been contacted, has been crisscrossed with each other. See the point at the center, you can see the two alpha helix of the two monomers have been crisscrossed with each other to form dimerization domain. At that point they interact. With the help of their leucine amino acid. So leucine is actually observed at the dimerization domain, the point where two alpha helices crisscross with each other. Now, due to presence of leucine in the alpha helix, they are helping to zip the two alpha helices together by creating hydrophobic domains. Due to presence of leucine in the alpha helices, they are helping to zip means just like a zipper. They help to zip the two alpha helices together by creating hydrophobic domain. So due to the presence of leucine in the alpha helix, it is thought that they are helping to zip the two helices together by creating hydrophobic domain. So see, the two turns in the alpha helix, you can see here, they are interacting with each other. Okay, see the leucine, the seventh amino acid, they are interacting with each other with the help of hydrophobic, with the help of creating a hydrophobic domain. Actually, this hydrophobic core region will interact by the help of hydrophobic amino acid. So this hydrophobic core region will interact by the help of hydrophobic amino acid. Rest of the amino acid outside are hydrophilic, which can interact with only water and other molecules, means other solutes. So due to the presence of this leucine in the alpha helices at the 7th amino acid, leucine is found at every 7th amino acid of the alpha helix. So these leucine are helping to zip the two alpha helix together, just like a scissor is closing its two ends. Okay, so they are helping to, this leucine is helping to zip two alpha helices together by the help of hydrophobic interactions, right? And this hydrophobic core region will and this hydrophobic core region will interact with the help of hydrophobic amino acid. Obviously, rest of the amino acid outside are actually hydrophilic amino acid, which can interact with water and other molecules. So here it has been saying a cross section of alpha helix has been shown here, and what they are saying that hydrophobic amino acids present in the alpha helix, internal sides of the alpha helix, are interacting with each other to form the hydrophobic core, which helps in assembly of the two alpha helix together. Again I repeat, the hydrophobic amino acids which are present in the internal sides of the two alpha helix, they interact with each other to form hydrophobic core, which helps in the assembly of two alpha helices together. Okay, so this is about the leucine zipper. You can able to see that two monomers with having two alpha helices are crisscrossing each other to form this helix, and within the dimerization domain they have interacted with the help of their leucine, because leucine is found at every 7th amino acid residue of alpha helix, and they can able to zip by the help of hydrophobic interactions between the leucine and other hydrophobic amino acids. They can able to interact with each other by how, by the help of hydrophobic domains. They are creating hydrophobic domains. These hydrophobic amino acids are actually interacting with each other to form hydrophobic core, okay, which helps in assembly of these two alpha helix. Okay, the rest of the amino acid which are hydrophilic, they are interacting with the water outside, so it maintains stability, perfect stability. It will not be getting hampered. Okay, the leucine molecules are surrounded by region of basic amino acid. Leucine molecule, the leucine amino acid molecules which are surrounded by a region of basic amino acid, it has been shown that the sequence of the basic region shows a certain degree of conservation. So these leucine molecules are actually surrounded by a region of basic amino acids, the the, the positively charged amino acids, and it has been shown that the sequence of this basic region have certain degree of conservation among the species. Unlike other motifs, in this motif there is no direct binding of leucine zipper with the target DNA molecule. Other DNA binding motifs, they bind directly to the DNA, but here in the leucine zipper they do not directly bind with the DNA molecule, but it folds the protein in such a way that interactions of the basic amino acids with the side chain of DNA can occur. Again I repeat, very important, leucine molecules are surrounded by basic amino acids, and these basic amino acids are conserved among the species, right? Unlike other motifs, these leucine zipper have no direct binding to the DNA molecule. So what does leucine zipper do? The leucine zipper folds the protein in such a way, that the basic amino acids which surround with the leucine zipper, this basic amino acid can interact with the side chain of the DNA. Because basic amino acids are positively charged, it can easily able to interact with the negative charge of DNA molecule, right? So unlike other motifs, this motif has no direct binding with the DNA, uh, no direct binding of leucine zipper with the DNA. Actually, this leucine zipper folds the protein in such a way that your interaction of the basic amino acid, that I have just said, that leucine molecules is surrounded by basic amino acid, right? So that basic amino acid can able to interact with the DNA molecule. So these basic residues can form like a scissor, just like a scissor like structure, where the DNA molecules is trapped between the two arms of the leucine zipper. Okay, so if I show you, the DNA can interact between these regions, that as we can say. Okay, example of leucine zipper is actually many examples are given. I will okay, let us see about the examples first. Example of leucine zipper is your June phos, then yours CEBP. Okay, these are actually what? These are actually transcription factors. These are the family of transcription factors. If I say you about the Zun and phos are the transcription factors which are activated by your MAPK signaling pathway, that is your membrane associated protein kinase signaling pathway. Zun and phos are the transcription factors which are activated by your MAPK signaling pathway. In case of CEBP, C/EBP, they are actually what? C means a sequence, CCAAT, and EBP means enhancer binding protein. So CCAAT, enhancer binding protein, it is also a family of transcription factor. This CEBP is also a family of transcription factor. It is composed of membrane. It is composed of 6 members. The CEBP is composed of 6 members, naming from CEBP alpha to CEBP zeta, right? And these proteins, they are the members of the family of transcription factors, and they promote expression of certain genes through which they interact with the promoter, and they can recruit the activator, then which can alter the chromatin structure and allowing transcription to begin. So these are the examples of leucine zipper, that is your June, Foch, then your which are associated with MAPK signaling pathway, then your CEBP. Okay, okay. So now coming to this point, so this leucine zipper is composed of B zip family. that is basic region leucine zipper, because the interaction with the DNA is mostly done by the basic amino acid residues. It forms a scissor like structure where the DNA molecules getting trapped between the two arms of this leucine zipper. And I have also said that unlike other motifs, this motif has no direct binding of leucine zipper with the target DNA molecule, but it folds the protein in such a way, this leucine zipper folds the protein in such a way that interaction of the basic amino acids with the target DNA molecule can occur. So this leucine zipper comprises of your basic region leucine zipper. Leucine zipper has two broad structural importance that is very important. First, one, at the end of it, it has multiple leucine amino acid, because at this leucine residues will interact with each other by hydrophobic interactions. Leucine zipper has two broad structural importance. First is your one is at the end of it. It has multiple leucine. Leucine is occurring at every 7 amino acid residues of the alpha helix, and each of the alpha helix is made up of at least 30 amino acid, and out of those 30 amino acid, leucine found at repeated at every 7th amino acid residues. That means that it will be found at the 7th from N terminal, it will found at the 7th, then it will be found at the 14th, then it will be found at the 21st. In that way it will be found. So it has multiple leucine amino acid. These leucine residues will interact with each other by hydrophobic interactions. The region of leucine zipper that interacts with the DNA major group, this region has several protrusions that has arginine residues. Again I repeat, the region of leucine zipper that interacts with the DNA major group, the regions of the leucine zipper that interacts with the DNA major group, this region has several protrusions that has arginine residues. The arginine residues actually has been protrude out from the leucine zipper. As I have tried to show you in this diagram, arginine residues has been as denoted by ARG. If you can see, it is looking as any, but it is not any, it is ARG in the black ink I have written. So these arginine residues are protruding out from the leucine zippers. Okay, they are protruding out from the leucine zippers, and this will interact with your guanine residues of the DNA. The arginine residues which are protruding out from the leucine zippers, it will interact with the guanine nucleotide residues of DNA. This specificity of interaction will determine that these leucine zipper will bind to specific DNA sequence or not. So obviously guanine, where there will be mostly present, okay, the specific sequence, it is observing the specific sequence actually. So this region of leucine zipper that interacts with the DNA major group, this region of leucine zipper has protrusions where your arginine amino acid has been protruded out from the leucine zipper. This arginine will interact with the guanine residues. So this specificity interaction will, that arginine is interacting with the guanine, right, it will determine that leucine zipper will bind to specific sequence of DNA or not. So this was about the leucine zipper. Let me talk about the last one, your helix loop helix motif, the last one. In helix loop helix motif, an extended alpha helical region from each of the two monomers, here also two monomers are present, two proteins insert they, and an extended alpha helix is present also. So in helix loop helix motif, an extended alpha helical region from each of the two monomers insert into the major groove of DNA and interact with the DNA. Let us see if you can able to observe it clearly. So you can see two monomers forming the two alpha helix, just like leucine zipper it looks like, but it has a loop. It has a loop like this. These are the loop that helps to pack together, and these are the two alpha helices that are involved in DNA recognition at the major groove of DNA. The two alpha helices that are involved in DNA recognition at the major groove. So that is the thing they are saying, that in this helix loop helix motif, an extended alpha helical region from each of the two monomers, an extended alpha helical region from each of the two monomers insert this alpha helical region, this extended alpha helical region. As you can see, this extended alpha helical region of these two monomers insert and interacts with the major groove of DNA and interacts with the DNA. The dimerization surface is formed by two helical regions. First part of the same helix is involved in DNA recognition. It is meaning to say that the first part of the helix involved in dimerization, and that same part of the helix, that helix there have a second part. This second part is involved in recognition with the DNA. The first part, which is a shorter alpha helix, it is involving in dimerization. The second part is involving in recognition. First part, the shorter part of the alpha helix involved in dimerization with each other to form dimer, and the second part of the alpha helix involved in your recognition with the DNA. Okay, so this so what I have said, the dimerization surface is formed with two helical regions. The first part of the same helix is also involved in the DNA recognition. The other is a shorter alpha helix, right? The other one is a shorter alpha helix. So you can see a shorter alpha helix, a longer alpha helix. The longer alpha helix are involved in dimerization as well as in your recognition with the DNA. The shorter alpha helix are what? The shorter alpha helix also helps in dimerization. So these two helix are actually, the shorter alpha helix and the longer alpha helix are separated by a loop. They are separated by a flexible loop. As you can see here, a flexible loop is present between the two alpha helices, both in these two monomers, okay, that is separating. So the two helices are separated by a flexible loop that helps them to pack together. So again I repeat, I think I have said something wrong. Okay, I'm trying to rectify my mistake. So I have said that so leucine, this helix loop helix motif have actually what they have, they are made up of two monomers, right? And these two monomers having the alpha helix which interacts with each other to form a dimer, and these two monomers, their alpha helices are interacting with the major groove of the DNA, right? Okay, second, I have said something wrong. That is, they have a two alpha helix, right? A short alpha helix and a long alpha helix. The long alpha helix are both involved in dimerization as well as for recognition. If you see the long alpha helix, the first part, the first half of the long alpha helix helps in dimerization, and the same long alpha helix of the two monomers are in the second half of these two alpha helix are involved in recognition. Okay, while the short alpha helix are found on the outside surface joined by a flexible loop, okay, that helps to pack together. So the dimerization surface is formed by two alpha helical regions. The first part of the same long alpha helix involved in DNA recognition, and the upper part involved in dimerization, right? while the another there is also a shorter alpha helix, and these two alpha helix are joined by a flexible loop that allows to pack them together. Leucine zipper and helix loop helix motifs are also called basic zipper or basic helix loop helix motif. Why? Because the recognition of the region of alpha helix that bind with the DNA contain basic amino acid. Again I repeat, a short question may come that why leucine zipper and helix loop helix motif are called basic zipper and basic helix loop helix motif, because they have basic amino acids with the help of which they can interact and bind with the DNA. Okay, so this was about the helix loop helix motif, and this was all about the motifs that I have talked to you in this video. I hope you can able to understand this. Okay.